Introduction. Welcome to today's lecture on the fascinating field of biochemistry, focusing on various aspects of enzyme catalysis. Enzymes play a pivotal role in numerous biological processes, including drug metabolism, DNA repair mechanisms, signal transduction pathways, and many more. Understanding the intricate workings of these enzymes is crucial for advancing our knowledge in both basic and applied medical sciences. In this lecture, we will explore 12 intriguing topics related to enzyme catalysis, delving into their mechanisms, functions, and implications in various biological contexts. So let's dive into the world of enzymes and uncover the mysteries of their extraordinary abilities. Enzyme catalysis in drug metabolism is a pivotal process that determines the fate of pharmaceutical compounds within the human body. Specifically, drug metabolizing enzymes, such as cytochrome P450S, glucuronidyltransferases, and sulfotransferases, facilitate the biotransformation of drugs into more soluble metabolites. This metabolic transformation enhances drug elimination and ensures optimal drug efficacy and safety. The mechanisms underlying drug metabolism involve drug recognition, binding, enzymatic transformation, and subsequent elimination. For instance, cytochrome P450 enzymes utilize oxygen and various functional groups, such as hydroxyl, amine, and carboxyl, to modify drugs through oxidation, reduction, and hydrolysis reactions. These processes not only facilitate drug clearance but also influence drug-drug interactions and the development of drug resistance in certain cases. Understanding the complex interplay between drug structure, enzyme activity, and patient-specific factors is crucial for effective drug design, dosing, and therapeutic management. In this video section on enzyme-mediated DNA repair mechanisms, we explore how enzymes play a crucial role in maintaining the stability of our genetic material. DNA is constantly exposed to damaging factors, but specific enzymes have evolved to recognize and remove damaged bases through base excision repair, BER. Other enzymes collaborate to synthesize and replace the excised base. Nucleotide excision repair, NER, removes bulky DNA lesions caused by harmful agents. Understanding these repair mechanisms allows scientists to develop strategies for preventing genetic disorders and reducing the risk of cancer. Metalloenzymes are enzymes that use metal ions like iron, copper, zinc, and manganese to perform their functions. These metal ions act as Lewis acids and help in transferring electrons or functional groups in enzymatic reactions. Metalloenzymes are vital in processes like oxygen transport, electron transfer, and redox reactions. They also play key roles in cellular respiration and the citric acid cycle. Metalloenzymes help maintain redox balance and can be targeted for therapeutic interventions. Understanding their chemistry and structure can aid in designing synthetic analogs and inhibitors for treating diseases. Ribosomes are RNA molecules that have catalytic activity, challenging the notion that enzymes are only proteins. They play a role in understanding the origin of life and biochemical reactions. Ribosomes can cleave, ligate, and polymerize RNA without protein cofactors. They have revolutionized the RNA world hypothesis and highlighted RNA's role as both genetic material and a catalyst. Examples include hammerhead ribosome, which cleaves RNA and regulates gene expression, and group I intron, which catalyzes self-splicing reactions. Studying ribosome structure and mechanisms can lead to therapeutic strategies and artificial RNA enzymes. Signal transduction pathways are complex networks that enable cells to respond to external signals. Enzymes play a critical role in these pathways by transmitting and amplifying signals, regulating cellular processes, and coordinating responses. For example, protein kinases and phosphatases catalyze the reversible phosphorylation of target proteins, an important mechanism in signal transduction. Enzymes like G-protein-coupled receptors and adenylal cyclases generate second messengers, such as CAMP and cGMP, which regulate cell functions. Understanding the interactions between enzymes, second messengers, and downstream effectors is vital for targeted therapies in various diseases. In this video, we will explore the fascinating world of Proteuses. Proteuses are a diverse group of enzymes that break down proteins in our bodies. They are involved in essential cellular processes, regulating signaling pathways, and maintaining our immune response. Proteuses are incredibly specific, targeting specific sequences of amino acids and proteins. We have hundreds of proteuses in our genome, each with unique functions and mechanisms. Some proteuses, like trypsin and chymotrypsin, use serine residues to break peptide bonds, while others, like papain and caspases, use cysteine residues. Metalloproteuses, meanwhile, rely on metal ions, like zinc, for their catalytic activity. Understanding the roles of proteuses is crucial for developing therapies and diagnostic tools for diseases like cancer and neurodegenerative disorders. Antibiotic resistance has emerged as a significant global threat to public health, necessitating a deeper understanding of the enzymatic mechanisms that confer resistance. Microorganisms employ various enzymatic strategies to counteract the effects of antibiotics or to modify them for their own benefit. These mechanisms include antibiotic modification, target site alteration, antibiotic efflux, and enzymatic degradation. For instance, many bacteria possess beta-lactamases, enzymes that cleave the beta-lactam ring common to several classes of antibiotics, rendering them inactive. Additionally, some pathogens modify antibiotic targets, such as DNA gyrase or ribosomes, causing decreased binding affinity and decreased susceptibility to inhibition. Furthermore, efflux pumps can actively transport antibiotics out of the bacterial cell, reducing intracellular antibiotic concentrations. By understanding these enzymatic mechanisms, scientists can design novel antibiotics that are less prone to enzymatic degradation or develop strategies to inhibit antibiotic resistance enzymes, thereby combating the alarming rise of antibiotic-resistant pathogens. Enzyme engineering involves modifying enzymes to improve their efficiency, stability, and specificity for different applications. This allows for advancements in industry, pharmaceuticals, and environmental cleanup. Two methods, directed evolution and rational design, are applied to engineer enzymes. Directed evolution creates diverse enzyme libraries through genetic mutations, selecting for variants with improved properties. Rational design involves computer modeling and structural guidance to specifically mutate enzymes. These approaches result in enhanced biocatalysts for chemical synthesis, pharmaceutical production, and waste treatment. Enzyme engineering maximizes their potential for bio-based applications. Plant secondary metabolites are diverse compounds that serve various ecological functions, including defense against pathogens, attraction of pollinators, and allelopathy. Enzymes play a central role in the biosynthesis of these unique plant compounds, providing the key reactions and chemical transformations necessary for their production. For example, terpenoid syntheses are a family of enzymes that catalyze the cyclization of the precursor molecule, isopentenyl diphosphate, IPP, generating an array of terpenoid compounds, such as essential oils, sterols, and carotenoids. Similarly, phenylpropanoid enzymes, such as chalcone synthase and stilbene synthase, facilitate the synthesis of flavonoids, lignin, and other phenolic compounds. These specialized enzymes exhibit exquisite substrate specificity and orchestrate complex enzymatic cascades, leading to the production of diverse secondary metabolites. By unraveling the biosynthetic pathways and understanding the enzymatic mechanisms involved, scientists can harness the power of plant enzymes to develop novel natural products with pharmaceutical, agronomic, and nutritional significance. Coenzymes and cofactors are non-protein molecules that tightly associate with enzymes, playing essential roles in facilitating catalysis. These molecules function as carriers of specific chemical groups or electrons, providing active sites for enzyme-substrate interactions. One prominent example is NAD+/NAD, a coenzyme involved in numerous redox reactions. NAD+ functions as an electron acceptor, while NAD acts as an electron donor, enabling the transfer of electrons and hydrogen atoms during enzymatic reactions. Similarly, other coenzymes, such as biotin, coenzyme A, and flavin adenine dinucleotide, FAD, participate in various enzymatic reactions, including carboxylation, acyl transfer, and oxidation reduction reactions. Metal ions, such as magnesium, zinc, and iron, also serve as cofactors, modulating enzyme structure, stabilizing enzyme substrate complexes, or facilitating specific catalytic steps. Understanding the roles and mechanisms of coenzymes and cofactors in enzyme catalysis is crucial for deciphering metabolic pathways, designing enzyme inhibitors, and developing therapies targeting specific enzymatic processes. Oxidative stress arises from an imbalance between the production of reactive oxygen species, ROS, and the cellular antioxidant defense mechanisms. ROS are highly reactive molecules that can damage cellular components, including proteins, lipids, and DNA, leading to cellular dysfunction, aging, and the development of various diseases. Enzymes play a pivotal role in maintaining redox homeostasis and counteracting oxidative stress. Superoxide dismutases, catalysis, and peroxidases are key antioxidant enzymes that scavenge and neutralize ROS or repair ROS-induced damage. Additionally, enzymes involved in DNA repair, such as poly-ADP ribose, polymerases, PARPs, facilitate the removal of damaged DNA, minimizing oxidative damage-induced mutations and genomic instability. Mitochondrial enzymes, such as the mitochondrial electron transport chain complex enzymes, also play crucial roles in cellular respiration and ROS production. Understanding the intricate enzymatic networks involved in oxidative stress response and aging is crucial for developing strategies aimed at preserving cellular integrity, preventing age-related diseases, and promoting longevity. Enzyme dysregulation is a key feature of cancer, impacting cell growth, survival, and spread. Many enzymes are involved in different stages of cancer development and can be targeted for treatment. Kinases, like EGFR and HER2, are crucial for tumor growth and can be inhibited using small molecules or antibodies, leading to better patient outcomes. Proteolytic enzymes, such as MMPs and UPA, contribute to tumor invasion and can be targeted to prevent metastasis. HARP1 is another enzyme of interest for cancer therapy. Understanding enzyme dysregulation and developing targeted treatments has the potential to greatly improve personalized cancer care. In conclusion, enzymes are vital catalysts that drive biochemical reactions in living organisms. This lecture explored diverse topics related to enzyme catalysis, such as drug metabolism, DNA repair, and signal transduction. Understanding enzyme complexity helps us gain insights into biological processes and develop innovative therapies. Enzymes hold the key to understanding life and offer opportunities to improve human health. Let us continue to explore the fascinating world of enzymes and uncover more secrets to further our understanding of life itself.